Welcome to part 2 of All Things DNA from Woodside Academy. Last time we actually built a DNA molecule piece by piece using the nucleotides adenine, thymine, guanine, and cytosine, as you see here. When you build a DNA molecule, it has two sides, so it comes out to be a double helix. But what does DNA do anyway? Everybody makes such a big deal about DNA, but what does it do? Well, actually, frankly, absolutely nothing. What it is like a cookbook of recipes. A cookbook of recipes doesn't do anything either, but all the recipes are there so that you can cook what are written in the recipes. So DNA is really just a cookbook of codes. So how do we actually use DNA and what does it turn into? DNA actually will ultimately turn into proteins like enzymes and body parts. And so everything that you are and everything that you're able to do is pretty much coded for in your DNA. So to get started, we take DNA and we have to open it up, okay? Because it's all stuck together there with the A's and the T's and the C's and the G's. And so we got to get at the codes. We're going to use this enzyme, RNA polymerase. And that's going to split our nucleotides right up the middle, right between their little hydrogen bonds. Now what we have to do is make an RNA from the DNA. Now RNA is just a little bit different than DNA. It's different right over here. There are no thymines, these little green pieces. There's no thymines in RNA. There's uracils. So every place that you see an adenine, instead of putting a thymine there, you're going to have to put a uracil there. And so what are we really doing, though? We're building an RNA copy of the code of DNA. DNA can't leave the nucleus. It's just too precious a molecule. We can't let it get destroyed, because if it gets destroyed, then you'd be all messed up. You'd be a true mutant, and we wouldn't want that. So we have to keep DNA all up in the nucleus, all safe and sound. So we make this coded copy of the DNA, and it's called messenger RNA. It's not DNA just because of the uracil. Whoops. One of my cytosines got away from it. Okay, and then here we go, and finally we're going to finish our messenger RNA, and it's all done. Now we're still in the nucleus, right? But now we're going to actually leave the nucleus with the messenger RNA. The DNA stayed behind, it's still safely tucked away in the nucleus. But now our messenger RNA is out in the cytoplasm, and what you see it attached to, this big blue thing right here, this is a very special organelle called a ribosome. And a ribosome is a place where the messenger RNA is going to get decoded. Every 3 letters is important. It's like a 3-letter word in DNA language. So like cat, dog, rat, except we have words like ugh, cug, whatever. Okay. So if you look here, we have A, U, and G. Well, A is going to match up with U is going to match up with A, and G is going to match up with C. So where do I find that? Oh, right here, UAC. Now what is this little devil coming over here? He is a transfer RNA. The transfer RNA is going to match up, same little coding trick that we did before, with the messenger RNA at the ribosome, but he's going to transfer or drag with him this precious little gem over here called an amino acid. If you remember, amino acids are the monomers of protein. I told you DNA had the codes for proteins, so we're going to put these amino acids together and we're finally going to make a protein. So if I match up, C goes to G, U to A, G to C, and now we have another amino acid that's going to get attached to the first amino acid. Now we have 2 pieces for our protein. A binds to U, C binds to G, C binds to another G, and we have a 3rd amino acid in our protein. And then finally, when all proteins are done, a release factor comes in, stops protein synthesis, and you have a protein that will drift away. Well, there goes our ribosome. All right. There's our protein. And away goes our messenger RNA. And actually, the other parts actually all get recycled, but here's our protein, and we're all finished with it. Now, this protein only has 3 amino acids. The average protein has 200 amino acids, which means if you need 3 pieces of DNA for every 1 amino acid, the average code for a protein in DNA is 600 amino acids. Next time you feel sorry for yourself, remind yourself you don't have to do that for homework. This is our second segment of DNA. I hope you understand protein synthesis now. If not, go ahead and stop it and replay it again.